Within the science community, it's fairly common knowledge that the entire human genome has been coded. Each of the 3 billion base pairs have been noted, all to push forward the progress of human medicine. Started in 1990 and completed in 2003, the Human Genome Project set out and succeeded to code the entirety of the human genome. But, as we know now, the genome consists of billions of base pairs. So, how exactly were they able to code them? Simple. They used Sanger sequencing. In 1975, Fred Sanger developed the first method to code DNA sequences. Sanger was a prominent figure in the science community, holding a position as head of the division of protein chemistry at Cambridge. Sanger determined the amino acid sequence for insulin, after which he turned to the chemical makeup of DNA and RNA. He started with methods for sequencing small portions of RNA. These methods were later developed to be used in sequencing the human genome. So, how does Sanger sequencing even work? DNA is first extracted from the cell through basic DNA extraction process, breaking the cell open, lysing the proteins and other contaminants, and cleaning the sample. This now purified DNA sample is put through the PCR process, which, through cycles of heating and cooling, the DNA can be replicated. Unlike normal PCR, however, normal base pairs, DNTPs, are mixed with chain terminating pairs, DDNTPs. DDNTPs lack a 3' hydroxyl end, which prevents any more base pairs from being added. There is also a fluorescent tag with a color pertaining to the specific type of base pair added: adenine, guanine, thymine, or cytosine. This allows DNA fragments of all lengths, differing by a single base pair, to be formed. Because the matrix of an agarose gel is too large to differentiate the DNA strands by their single base pairs, a polyacrylamide urea gel with a tiny matrix is used. Capillary electrophoresis is used to separate the DNA strands, and a laser is used to light up the fluorescent tag so the gel can be read. Because the smallest strands travel the farthest in the gel, these are read first, as these would be the first base pairs in a DNA sequence. The data is generated as a chromatogram, which has colored peaks according to the base pair's fluorescent tag. The colored peaks are red, which gives the sequence of the DNA. This method was used to code the entire human genome through a painstaking process. However, those 13 years were well worth it, as new innovations rose in the medical community because of this coded genome. While time consuming, the Human Genome Project boosted scientific innovation, all thanks to Fred Sanger and his sequencing.